he is so famous, so I don't think he require my introduction, but still I would like to give some of a little bit brief intro about Professor Vijay Ram. So he is a professor of chemistry. He is coordinating the supercomputing facility for bioinformatics and computational biology at IIIT, also at IIT Delhi. So a little bit about his academic journey. So he joined IIT Delhi as a faculty in chemistry department back in 1990, and he obtained his PhD in chemistry from the City University of New York in 1986. Soon after that, he also got his postdoctoral training with Professor Barry Honig, so one of the pioneers in the field of biomolecular electrostatics and bioinformatics at the Columbia University in the US. So Professor Jayaram contributed a lot in the case of protein structure predictions, drug discoveries, and all that. Everybody knows that. I just want to highlight some of the key things like which I think whole of the world is using from his lab, which is basically the part of what he quoted as the gene to drug software suites. So it includes the genome analysis programs, which is called it as ChemGenome, and Bhagirath, I think everybody of us know very well. We used it. He also contributes, lab contributed to Sanjeevni, and there are so many different softwares, open source softwares coming from his lab, which are widely used. He has published, he has one of the stellar series. He has published so many great articles in high impact international journals. He has, multiple PhD students. He has guided them, and so far and so on. I mean, if I keep on talking, it will never end. So I leave it to you, Professor Jayaram, eagerly waiting to listen to you.
Oh, thank you. Thank you, Professor Garo. Thank you very much. And thank you, Professor Raghav, for this opportunity. It's wonderful. I see fantastic list of speakers. It's very heavily crowded. I think it looks like a regular full-scale conference. It's wonderful. I'm so happy that you managed to organize it. Looks like now Professor Chandra will probably be very annoyed if I don't finish it by 10:30. So I think looks like I have 30 minutes now to finish the talk. So let me do it, justice to it. Meanwhile, I'll share my screen. Let me turn off my video. Is this visible to the audience?
Yes.
Yes. Yes.
Ah, wonderful. So this is a full screen mode, right? So, okay. All right. So let me start. So I'm calling this as Dhanvantari. This is more like an update. I've been talking about it for a while now. So, and there's some new additions to this and some additional experimental confirmations of the process here. So coming back to our dream plan, the dream plan is someday maybe contributions coming from AlphaFold 2, as our chairman was asking a little while ago. Someday, maybe as a disease is reported at 10 a.m. and the drug is delivered at the doorstep by Amazon or Zomato at 5 p.m., right? That is a general plan. And maybe now 20 years from now, this will happen. And with heavy inputs from machine learning, and I'm sure this is feasible. Right now, we're saying genomics, proteomics, metabolomics, information technology, but we should probably put this artificial intelligence somewhere out here in addition to the chemistry, right? And the urgency, everybody knows, we are becoming very complacent with regard to new molecule discovery, especially antibacterials. Our discovery stopped in 87, but bacteria are becoming smarter, and because of the AMR, antimicrobial resistance, now there is an urgency to develop some new molecules. And this pandemic is also kind of insisting that we should do something about antivirals to not only antibacterials, right? Pathogens in general, right? But we've been blaming on the R&D being very expensive. How expensive it is? Well, 2016, they revised it to $2.6 billion in 14 years. And now, of course, this is only going up. In 2021, I would presume that this is probably touching $3 billion. What we expect is in silico interventions, and we should cut down the time and cost. The previous speaker has given ample justice to this theme of cutting down. So I would just say, "Aapka kya hoga, Janabe Ali," right? And of course, what are we trying to do? Well, we are trying to use this data and then come up with a genome annotation method. There are several of them. I will mention as to what is this ChemGenome, why another software, why one more software here, why another software. But overall pathway is that from genome, you identify protein coding regions using this chem genome. Chem genome can do much more than protein coding regions, but for now, let's say we got a little piece of DNA. Writing from DNA to RNA is very easy to write. And from here, Rnas code gives you the polypeptide sequence. The challenge, of course, is to go to the 3D structure and then on to a small molecule. Most of the drugs in the market are targeted to proteins. There are some molecules towards nucleic acids, microRNA, for example, or the DNA, it's binding drugs for cancer, anti-cancer molecules, but 90% of them are towards the drugs. And so this is in the language of Professor Balwant saying, so it's just like structure-based drug design. It's where you can have a molecule towards a DNA or towards a protein as an inhibitor again, not as an activator as yet, because we still haven't figured out a, a, a certain pathway, a clear pathway to come up with activators. But someday, sure, will happen. That will also happen. So coming back to this question, does this Dhanvantari really work? No, basic question. Now, this is again a colleague of mine in the School of Biological Sciences. So he gave me a challenge. He is the world's expert on hepatitis B virus. And he says, give me a new molecule, and he says something about this is already known for WRFCR proteins and their function. Everything is known already, and there are a few FDA approved drugs as well. Most of these drugs are nucleoside analogs. I'm sure some of you know that AGCT, right? They are nucleic acid bases. You put a sugar, they become nucleoside. You attach a phosphate, then they become a nucleotide. So most of these drugs are nucleoside analogs, and the virus has developed resistance to these nucleoside analogs. And what are these nucleoside analogs doing? Well, these are all targeting the viral polymerase. So his question to me was, this is my colleague Vivek's question was, can you target another protein, not polymerase, and give me a molecule that is not a nucleoside, right? Then I told him, give me $2.6 billion and 14 years, I'll definitely get to a drug molecule, right? That was, well, more seriously, just give me 3 hours, I will do it. Well, how do you do a molecule in 3 hours? Well, remember our dream plan. Don't stay away from the dream plan. 10 a.m. the disease is reported, 5 p.m. the drug has to come to your doorstep, right? So how do we do it? So I go to ChemGenome. This is our software from SEO Bio. This is freely available. I can download this hepatitis B virus genome from the NCBI site. Again, this is free. And once I do it, I push it to this ChemGenome software. It identifies 5 protein coding regions. And these 5 proteins do not share any similarity with human proteins. So that is good news. So you could probably target any one of them. But my friend said, don't touch the polymerase. And he was particularly keen that I should focus on a surface protein, etc. I said, okay, we'll take surface protein, but we could have taken any one of them or all of them, right? So we now have a sequence from here. So we have a protein coding sequence, and the protein looks like this: methionine, glutamic acid, asparagine, isoleucine, etc. So this is a protein. Now I push this to Bagheera software. It gives me a 3D structure, 3-dimensional structure. Once I have this, I start looking for binding pockets here, 1, 2, 3, 4, 5, etc. And after I get it, I identify about 10 binding pockets. Remember, I'm not putting in any experimental information.
अभी भी आप बेसिकली डाउन नहीं, हाँ, तो आप अपने आप को डाउन नहीं करना है, आपको अपना बेस्ट देना है.
Oh, okay. I think Professor Ragwa has some announcement, I guess. Okay, so let me continue. So as of now, we have not done any experimental input. So we started with the genome, from genome annotation to protein, and from protein to sequence to structure. And now we are looking at the structure, butane binding pockets on this using this ASF. This is an active site finder of Sanjeevani software. Sanjeevani is our drug discovery software. So, and then I move on to my Rspd. Now, in 2020, that is last year, we have come up with a newer version of this Rspd. We call it as Rspd Plus. This is in collaboration with my German colleague, Professor Rebecca Wade. And so that seems to do a slightly better. So that is also freely available. And it's downloadable too. You can download it onto your laptop and work with that Rspd. What is the goal of this? Well, you can scan 1 million compounds in a matter of seconds, and probably seconds is an exaggeration, maybe minutes. Like in 10 minutes, you can go through 1 million compound library based on a QSR type equation. And that is, so that essentially gives you a smaller set of molecules to deal with for the purposes of atomic level docking and scoring. So the output of this Rspd of the Sanjeevani is about 150 molecules. based on a threshold. So we put that, so from 1 million, we've come down to 150 molecules. And then we do atomic level docking using our Paradox software. This is a parallel docking software. So it takes like 20 minutes on about 8 processors, and we do 10 such docking simultaneously. So about 80 processors are occupied for about 20 minutes, and then you get decent structures out of it. People have said, well, there's an Autodock, why Paradox? But I mean, in our experience, a Paradox seems to do a better job than Autodock in terms of RMSDs when in relation to the crystal structures, but it depends on the users. So this may be a subjective statement, but we have some numbers to validate that idea that Paradox freely available software is just as good, if not better, than most of the existing docking softwares in the world. So we now have a few molecules. So 30 molecules here ranked according to the binding free energies. And so these are all zinc IDs, etc. And then I said to my friend, well, take these top 5 molecules. So our job is done, basically. So this is done in 3 hours, remember? So we started with the genome. Now we have 5 molecules. And now the job for Vivek is to test them in an experiment. Now, what does he do? Well, he finally does a lot of testing. This testing has been going on for 3 years, and the referees wouldn't let him publish this paper till he finished all possible biologies. Finally, it got accepted recently. So we are very excited. And in fact, one of the molecules, that molecule 5 that I was showing you here, it's a piperazine derivative. This seems to work very well. And in fact, it shows low micromolar. And then interestingly, it also works on mutant strains. More recently, this like last week, in fact, he was asking me that he has tested this molecule on 8 other mutants. And he says, I want to publish it. I want to put you as a co-author. I said, why? I didn't give anything to you. You did all the biology, and you should just go ahead, put me in the acknowledgement. No, he was asking me that if this computer didn't help us, we would not have known that this is the zinc 377 molecule out of the 1 million molecules that needs to be tested against the mutant virus. And so that's a good success story. And I'm sure I'm trying to say that CADD or computer aided drug design is working great, but most of you are already sold out. I'm sure you know as much as I do that this is a useful tool to do further testing. So in my opinion, this is great, that we have a predicted molecule and it's working experimentally, and virus clearance is done, all kinds of tests were done. But what is missing is an input from toxicology. If only there was in my protocol one toxicology module, I would say, well, it's fantastic. That will be really good. So I can get you hit molecules, from hits to leads to drug molecule or druggable molecule. So that is a challenge still. So that little toxicology part is missing, but I'm hopeful with machine learning inputs and with enough data, we should be able to conquer and get the molecule that we want within a matter of a few hours, right? So now for some quick challenges, scientific challenges that are out there along the pathway. So focus on the science part, leave the software part. What is this? Of course, we call this genome. So for those of you who are not biologists, I'm just putting this at the end. Entire content, DNA content is a genome, and the protein content is a proteome, etc. Right? And genomic diversity explains, plus environment, of course, right? And this is a central dogma that I've been talking about: genome to the DNA to RNA, RNA to protein, protein to a structure, and structures are these nanobio machines, that are keeping us alive, and structures are our targets. What is the question in the genome? We use a lot of homology kind of softwares to predict ORFs, etc. But if you look at the sequence GATCTTTAGA, now what is the meaning of this word? Here is a nice word, 10 letters, right? Nobody knows. That is the unfortunate part, right? Here there is a GATAC, AAAAC, etc. This is a big challenge here. This is not like, twinkle, twinkle, little star, you can understand it. In fact, English language is so complicated, 26 letters. Still, we seem to be masters of English language, but this is just 4 letters and we have no clue what's going on. My feeling is that this man knows, except he's not telling us, right? Except him, probably nobody else, right? So that is a challenge. So the challenge is, if I don't give you this information, the definition as there in the NCBI, you wouldn't be able to figure out that this is a a naughty girl, or there's a handsome boy, etc. This is Y chromosome. So that's a big challenge. And there are several gene prediction softwares available, tools available. Most of them are based on data. You need good data, then you train your model, and or then you make predictions. But then if you trained it on one particular system, you go to another system, it starts to fail very miserably. So that's no good, right? So that's the problem. In fact, I would not want you to focus on the last two columns. So sensitivity, specificity, they're supposed to be 1.0, 1.0 for a great model, fantastic model. They are less than 1. That means there is room for some more research and some discovery. But look at the middle methodology. This is the methodology that people have been adopting. Markov model, discriminant pattern recognition, fantastic mathematical, sophisticated statistical tools are being implemented, but you need clean experimental data to train your model. Then you make your predictions, right? But then are they transferable across the species? But that's a big question. They are not transferable across the species. That's what this story is telling us. So what is the challenge then? The challenge is develop a universal model. It works every time, like Watson and Crick story. Watson Crick says A binds to T, G pairs with C. No matter what the organism, whether it's a rice or wheat or human or bacteria, wherever, right? It works everywhere. But here, these gene prediction methodologies are not working. So that's a challenge. So develop a universal model that works every time, right? So we started with this idea of developing a hypothesis-driven universal model. So we thought DNA wants to talk to us. It wants to talk badly through some parameters which we don't know. So let's speculate. Hydrogen bonding should be good because Watson Crick said hydrogen bonds are important, right? Stacking. Not many have said much about stacking. This is one on top of the other, right? So that is the stacking part. One above, pair below. So that's the stacking part. And then there are grooves. Proteins are recognizing the information in the DNA. So they must be playing some important role. In the absence of protein, solvent does the job of recognition. So you can call this a solvation, stacking, hydrogen bond, etc. So this was a story from 2006. And now I'll do a quick run to 2021 very shortly. So you can pull these numbers out of molecular dynamics simulations. You can also do a paper pencil calculation, and you will be as good as 70% accurate. With this additional simulation input, probably you'll be 90% accurate. This is an interesting story. I will tell you on some other day, but right now I will just say that hydrogen bond and stacking can explain this degeneracies in the genetic code. I'm sure some of you have worried about there are 64 codons, but only 20 amino acids, and some of them are coded by 2 codons, some of them are coded by 4 codons. Why this? This must have a lot of confusion. It's unnecessarily creating too much confusion. This is what Corona said, but not Hargobind Corona, not Corona, right? So how do we take care of this? Well, hydrogen bond stacking can explain it. I call this as a rule of conjugates. This is for some other day. Now I use this rule as well in the absence of solvation. So here is a story of CAM genome. So give me a sequence. I'll take a walk along the sequence in steps of 3 bases at a time, 3 base pairs, in fact, at a time. And I add up hydrogen bond stacking. And this propensity parameter is a solvation. In the absence of solvation, I use that conjugate rule kind of thing. Now, my expectation is that these properties should differentiate between various properties, various functional units on the DNA. So if it's a genome or if it's, so that's the general idea here. Now, does it really do this? Well, as you see here, the vector, I convert this into unit vector and plot it for 900 prokaryote genomes. So each one is one particular genome. So you can see this is clearly transferable across all the prokaryotes. We have extended it to 2000 prokaryotes. It works there as well, right? And once you have this, writing a software is the easy part. So you identify universal plane, and then any vector that's above is coding for a gene, below is not coding for a gene, right? So that's our basis of the CAM genome software. Now, quickly putting this CAM genome on the same plot as I shown you earlier, sensitivity, specificity are not bad at all. And this is a transferable model. And you can tweak this idea. Why only 3 parameters? Why not 5 parameters? You can say, well, it depends on what are the parameters which are important to the DNA, language of the DNA. That is what you need. What you want, of course, is a universal model that works every time, right? In fact, I'm showing you Arabidopsis. This is not a prokaryote, by the way, right? Although this method was tested by prokaryotes, so you could say, well, it seems to work even for eukaryotes, right? What about promoters? Promoters also show up quite strongly. Stacking energy, hydrogen bond energy, they separate promoter from the genes. Intron exon, very challenging stuff. Again, these black ones. This is from rice genome, by the way. Rice genome is a eukaryote, as you know, right? So these peaks are all exons and the valleys are all introns. So there's a clear distinction, right? So this is a story from 2010. Now, beyond that, what did we do more recently was to identify fingerprints for different kinds of RNAs. Why only mRNA coding for proteins? Why not tRNA? Why not ribosomal RNA or microRNA, small different variations on the ribosomal RNA, etc. And apparently all these things, like hydrogen bond solvation stacking, they seem to segregate these bunches. And this is data from 7.6 million RNA genes. So what you're seeing is the data from these millions of genes, and there's a nice separation in this space, which is great news. So this is like a fingerprinting based on physicochemical properties, which is giving you the strength of universality. Remember, we want a universal model, right? And now transcriptional stuff, I'm sure everybody knows. These prokaryotes, promoted sequences particularly, they are very promiscuous. Sequence-wise, they don't get conserved at all. If the color is the same vertically, then you have a conserved base, right? But as you see, there are all kinds of rainbow colors here, right? But if you go to these physicochemical properties, all these promoters that I have shown you, here I'm showing you 31 parameters. All these are structural as well as energetic parameters, and coming from 12 organisms, and the data from 16,000 TSS start slides. You see a nice peak here, either a peak or a valley for each of the parameters. So in sequence space, there seems to be no conservation, but when you go to this property space, there seems to be a very clear signature. So if you're trying to develop a machine learning model, my request to you is consider property space. You will be probably more successful than training things on a sequence level. So I believe DNA wants to talk to us, right? So this is an 2018 story. More recently, there's a few months ago, finally this intron exon thing got published. And this is again based on structural energetic parameters at the boundaries. And I'm sure all of you know intron exon, there is hardly any sequence conservation. So sequence space, if you do it, you get into all kinds of problems. But when you do it in property space, however, there are clear signals as to where is the start of the intron, end of the intron, etc. So that's a nice story. And this is coming from how many? This is from human genome, by the way. So remember, I'm not doing any training on a prokaryote or eukaryote, just applying all across to all kinds of species. And this is based on 300,000 start and stop sites. And it seems to work quite well, right? So I'm sure, I hope I have convinced you that we should go towards a universal model, right? And then use a structural property space or energy space. Then your machine learning methods will work wonders, right? What is it that we want to do now in DNA? What is the challenge? The challenge that remains here is read genomes like Harry Potter novel. There's so much fun out there, right? Rice genome, buffalo genome novel 3, human genome, another novel, prokaryotic genomes, eukaryote, so much. What do we know so far? We've been only focusing on this coding DNA. Non-coding part is a great interesting story, and this is only 3%. So what is the remaining 97% doing inside us is a big question we should all explore. We need to know about ourselves at a genomic level more thoroughly. Now coming back to quickly on the drug design part. So that was a story journey of the chem genome from 2006 to 21. Now about this protein part, I'll just do a quick run through here. If you are to ask what's a disease or a disorder due to, well, I think functionally speaking, it's a protein either lazy or it's overactive or it's missing altogether, right? Or it's got mutated. So some such thing is happening to the protein. So most of the drugs are inhibitors targeted at the proteins. But someday, with the advances in nanobiotechnology and drug delivery systems, DNA could also be a popular target. But someday, very soon, right? This is, of course, now challenge that AlphaFold 2 is taking. Now, the gap is huge, millions and structures are less, etc. Where we may run into problems with the machine learning approaches is that number of folds has saturated. So we are drawing data from crystal structures, and 1400 is where things have stopped in 2008. If you look at this graph, 1400 folds. After that, the no new folds were discovered. Either crystallographers are using the existing crystal structures, or they're probably not going for new folds at all. Something is happening, or maybe the universe of 14 structures is restricted to 1400 only. So this is something that you got to keep in mind when you're doing PDB data based training of the models, right? So keep that in mind that we have saturated last 13, 14 years. Not many new folds have come out beyond this 1400, which is not a great news, right? So, but sequences, of course, going in millions, because we don't have structures now. With good methodologies, we will have probably, this is for the rice, very few structures are available, right? Homo sapiens, again, very few structures. So we are hoping to see these things filled up. 100% structures are available using computational methods. So the big phi psi is a big question that Ramachandran, can you predict Ramachandran angles? We've given a sequence is a big question. It's been a challenge. Now, apart from the AI methodologies, can you create a simple intuitive model that works in a standalone mode on a laptop? That's the challenge that I am asking. I'm giving to myself, especially in the context of this new Google procedure. So we have created this Bagheera software, which works quite well with small proteins, and also at too low resolution, up to 5 angstroms, we can get a good structure. But beyond that, if others like Baker software, Zhang software, Roberta's, and Tasser, if they're doing better in the CAS, it's because they seem to have some slightly better refinement methodologies. We have not spent enough time. But interestingly, my question to you, or those who are interested in proteins, is the following. Protein folding unsolved, protein DNA recognition unsolved, protein RNA unsolved, protein recognition unsolved, drug discovery, protein small molecule recognition unsolved. Common factor is the protein. What is the problem with the protein? Why are they all unsolved? DNA structure, of course, double helix unsolved. It is solved based on Watson and Crick. And what about Linus Pauling? Linus Pauling's secondary structure of the protein. He said alpha helices and beta sheets based on hydrogen bonds. So apart from hydrogen bond ideas, we seem to be lacking in terms of new ideas, especially for solving the proteins part. So people have classified proteins in so many different ways. Somebody said there are at least 1,000 ways of classifying protein properties, etc. But do you really need all of them? If they were right, we should have solved all these problems. So something is going wrong with our understanding of the protein structures. I'm cautioning you. What do we know clearly? Well, we know sequence to structure, right? Dhanvantari has said it. This is a 1972 Nobel. What else do we know? We know Linus Pauling's story of alpha helices and beta sheets based on hydrogen bonds. What else do we know? We know oil and water don't mix. This is a thermodynamic hypothesis. We extend it to structure, saying that non-polar residues are inside, polar residues are outside. This is a structural interpretation of a thermodynamic phenomenon. Whether we are right or not is different. He didn't get a Nobel though. Ramachandran, of course, did not get the Nobel, but what he said was this phi-psi plot, only 15% is occupied. Rest of it is not occupied because of steric clashes. Right? All of you are aware of that story. But these ideas, the four ideas that Anfinson and Paulings and Kauzman and Ramachandran, they're just not sufficient to build a tertiary structure. So what you need are some newer ideas, like construct a higher order Ramachandran map instead of a single peptide, just take 3 peptides at a time, then probably for small molecules. This is a 2015 story. That is a convergent problem is what we are claiming in 2016. Right? And then more interestingly, do you really need 1,000 properties of proteins? Maybe you don't need them. If you ask a simple mathematical question, these mathematicians call this as a D3 symmetry of a triangle, 4 colors, 3 sides, how many possible color triangles? 4 times 4 times 4, 64 color triangles. But because of the triangular symmetry, you can rotate it by 120 degrees, you can flip it by 90 degrees, 180 degrees, etc. So you'll end up getting only 20 unique triangles. Now, this 20 has nothing to do with amino acids. If you don't pay attention to the mathematics. What is the mathematics here? This red color in the 20 triangles occurs only 10 times. Red and blue together occur only 4 times. And red, blue, green occur only once because there are only 3. Right now, let's make a hypothesis. Amino acids have evolved out of 4 chemical properties, not 1,000 properties, not like textbooks. Let's simplify the problem. A minimum of 1, a maximum of 3. Each property occurs only 10 times. 10 amino acids. Any 2 occur only in 4 amino acids. Now you look at any of the favorite textbooks that you have, Lellinger, Stryer, whatever you want. No textbook lists any properties which satisfy any of these rules, meaning that these rules are nonsense, or maybe our textbooks have not come up to a stage where we can solve the problems. Now I will show you alternative perspective, which is presence of sp3 gamma carbon. Exactly 10 amino acids have the property. Hydrogen bond vulnerability, exactly 10 amino acids. And how many of these two are together? 4. And that also 1 and 2 occur only 4, and that's what is KQRT doing. And absence of delta, exactly 10 amino acids. Now 1, 2, 1, 3, 4, 2 and 3, again 4. That's what is listed here. So we have an alternative view, and you need only 4 properties. Now let's see if we can do homology modeling with just 4 properties, not thousands of properties. Moment you do it, and this is a 2018 story, you push the homology modeling to a much higher level. So we modified it much higher level than the existing homology models of proteins. So this is a story of biochemistry 2018. So I'm calling this as a chemical logic. More recently, in 2021, my student who just finished her PhD, Amita, she uses 4 properties. She does a mask blast, and she predicts function of protein sequences to a much higher level of accuracy than the existing softwares. So my appeal to you is don't worry too much about thousands of properties of proteins. I think they have 4 properties. Then what is hydrophobicity? Then what is polarity, etc.? Well, these are compound properties, not primary vectors. Right? Primary vectors are only 4. You can combine them, you can create several other properties. That's what is happening. So this is a Bagheera software. Yeah, of course, you've seen it. This is ranked at 11, or 12, or 13. So, but if you go for coarse grain models like GDT, this is one of the scores that CASP people use it. It's like ranked 2 or rank 1, but suddenly drops to rank 7 if you want a high resolution model. Low resolution model, there is no issue at all. So that's what I was mentioning. So this is a 2018, 2020 last year, or because of the pandemic, my students have dispersed all over the country. So there was no competition from our side, serious competition from our side. So if you focus on 5 angstrom type structures, you will see Bagheera also, along with all these Baker's software and so on, Zhang software server and so on. So someday, I'm sure we'll get there using a simple model without a supercomputer on a laptop. I'm sure we'll get there very soon. That's the challenge that we are giving. And once you do it, then you can always create a computational PDB. Now I think my time is out over. Now I'll just run through the Sanjeevani various softwares, acquaint you with the existing softwares that are there. So this is overall strategy. We have databases. Let's see what are the databases. So you can do a million molecule, like I mentioned to you with the Rspd, or you can do non-redundant database of small molecule natural products. All these are available on the Sanjeevani site. You can draw the molecule, you can check the LPC compliance, you can do a quick scan with Rspd protocols, and then you can do optimizing the geometry, doing simple energy minimization, etc. Assign force field parameters, you can do atomic level docking scoring. But the DNA part, we can then regenerate an MD average structure of the DNA and then do a docking, essentially minor group binding and intercalations. Both are possible. Then perform MD and post-factor free energy analysis. That's the overall flow chart, right? And this is the MG, typical GBSA, PBSA type analysis for free energies, right? And you can use these ideas to see whether there are drugs which are binding to off targets, like this drug 14 is binding to target 1, target 3. So this is bad news, right? It can cause off target binding and side effects, etc. You can prevent this using CADD. Not only get the molecule of your choice, you can also ensure that there is no off target binding, etc. So Sanjeevani success stories are anti-cancer. This is one for breast cancer. When I say success story, I'm referring to a case where it's been designed on a computer, then synthesized in the laboratory, or bought from the market and then tested experimentally. So experiment numbers are what you're seeing here, right? So this IC50 2.27, etc. This is an experimental number. And similarly for the anti-Alzheimer acetylcholinesterase, this was the target. And the previous case, it was the estrogen receptor. So here it is estrogen cholinesterase. So it's not tailored to a particular disease. That is basically, it's a software suite that works once you have a protein target, right? DNA target as well, right? N-methyltransferase again is unique to malarial parasite Plasmodium falciparum, and therefore it is a good drug target. And so the synthesis was done in Jamia. SCF Bio, we did the design part synthesis done in Jamia, and biology was also done in the Jamia part. Then picornavirus, 3C proteases, etc. This is again the experiment. We bought the molecule and it did the biology and the School of Biological Sciences at IIT Delhi. The design was, of course, at the SCF Bio. And in fact, this particular virus, this 3C protease, is pretty much similar to this virus of the SARS-CoV-2, this coronavirus as well. So we have some molecules for that. So this is a February journal, got published in 2018. Later on, my student also did some work on the chikungunya as well. So that got published last year in virology department. So because we were very happy to see Sanjeevani going into nature with an antifungal. So this was a story from 2016 from my collaborator, Arthanari Hari Babu from Harvard Medical School. Right. So that's a mobile app. So for downloading while you're traveling in metro, you can do drug discovery using Sanjeevani. So I was appealing to you that focus on this T. Please give me 5 rules of toxicity so I can create a nice software. And then we automate the whole process of drug discovery so that molecules that we generate are not toxic, but they're eliciting the job of what they're expected to do. So I summarized you the work of this chem genome and this Bagheera, this active site finder, rapid molecule screening, atomic level docking, and several filters, etc. So this whole pathway finally got published with lots of validations, several validations last year in chemical engineering science. So quickly on the pandemic, so what we did was we got molecules from FDA approved drugs, molecules from isotin based molecules, because these were synthesized in IIT Delhi Chemistry Laboratory, and they worked very well for Picorna 3C virus. So they're Indian medicinal plants. In this we in fact this is available, the resource is available at SCF Bio. We call it as BIMP, bioactivity information for medicinal plants, Indian medicinal plants. And so we now have named turmeric and tulsi. We're adding giloy, we are adding a few more molecules. So molecular structures or IDs, everything will be available. And this is easy research for docking. You can do a nice docking with the molecules. So that, as well as zinc compounds, so millions of molecules are available. And so we have identified a few. Now, biology part is a difficult part because for this SARS-CoV-2 virus, you need a BSL-4 lab. There are very few. Most of them are busy with developing kits, testing the kits, and testing the vaccines, and so on. So someday, hopefully, when they're free, they will start testing the small molecules, and we'll have small molecules sold across the counter in medical shops, so that this COVID pandemic is gone, right? It will be like any other viral fever. Very soon, hopefully, we'll have a tablet. So that summarized you our PhD work of 25 PhD students so far. So lots of storage and dedicated student power that has available. So these are all the various people who contributed to these Dhanvantari softwares. Thank you very much for your attention. Visit us. If you have any questions, please email me. Feel free.
So, I mean,